Thank you very much for this incredible introduction, and thank you for having me here today. It's really a great pleasure to speak in front of such an audience. So in this talk, I'm going to show you something on some aspects of the evolution of genes involved in the metabolism of foreign body compounds and drugs among them. Here you have this picture. You see this is the red queen, and I'm going to refer to this hypothesis that was originally formulated to explain in a framework of different evolutionary aspects, like evolution of sex or prey-predator cycles, just because it really fits to our system in a way that the gene we are talking about are involved in the transformation of compounds coming from the environment. Therefore, they have to co-evolve with changing environment. And in a way similar to what the red queen is doing, they have to keep on running to be in the same place. In other words, to keep the adaptation value high, so the fitness it should be high. Saying that, I'm going to kind of discuss two different approaches that we can use to try to understand something, to shed some light on the evolution of pharmacogenetics genes. The first one is concentrating our attention and start having hypotheses on the evolution of single genes. And I will call it the first approach. And then we will see something on how to approach the evolution of multiple genes together, analyzing their variation as if they were a panel of Snips or single sequences. So let's see here. You see this, the gene you have heard just about in the previous talk is CYP2D6, and why we are concentrating our attention on CYP2D6 when we are interested in the evolution of single genes. Because for population geneticists and people interested in evolution, this is a kind of a dream system, because like Scott said, there are more than 100 alleles, and so there is a huge amount of variation we can play with if we want to infer some evolutionary aspects. And you can see here that is also very interesting from the pharmacogenetic point of view, because it has several drugs are metabolized by this protein. So this is a network of CYP2D6 haplotype that we could infer when we studied 12 informative positions, the duplication and the deletion of the gene. What is a network? A network is a phylogenetic representation of the relationship between different haplotypes. And here you can see that each one of these circles is an haplotype, and let's say the bigger is the circle, the higher is the frequency of that haplotype. And you see a lot of colors that are referring to different populations. So what is this network telling us is that first of all, there are several haplotypes kept at a relatively high frequency in different populations, despite some of them being low functional haplotypes or even null functional haplotypes. This is not like an unusual thing to be for a coding gene. And plus, we also have this ultra-rapid haplotypes that are due to the fact that this gene has copy number variation, as we just heard. So lots of haplotypes, different phenotypes, and they are all represented in many different populations. So lots of variation, and this, in terms of evolution, opens several questions. For example, we can ask if the presence of so many slow function haplotypes distributed in several populations is due to convergent adaptation, or if there is a sort of relaxation of selective pressure. To test, first of all, if we want to infer something on the evolution of genes, we have to have an hypothesis. This is something that is very interesting for genes. This kind of Neolithic transition can be a sort of evolutionary framework in which we study how this gene evolved, because they, like I said in the introduction, they have something, they have a function that is related to the environment. And here you see, this is a very clear picture where you can see that our species went through this divergence at a certain time. Here you have the time frame, and it went first, this transition to agriculture led to a strong bottleneck at the beginning due to maladaptation, but then the introduction of this important new dietary compounds led to a strong population expansion. And this left a signature on our genome. You can see here, for example, the case of this gene that is metabolizing starch, and you see that this gene has a copy number variation, so the higher the number of copy, the higher the metabolic activity. There is a correlation between the two things. And you can see here from this picture that populations that have a high amount of starch in their diet, they have a higher amount of copy numbers, which means that here probably there has been a positive selection for a high copy number due to the fact that this amount of starch was beneficial and has a positive effect on fitness. But probably the most the most known case of positively selected genes in terms of Neolithic revolution is this lactase gene that you all know very well. This is the distribution of the lactase persistent trait, and you can see here that is almost fixed in some population, and it is clear that it arises due to convergent adaptation. So clearly having a strong impact on the fitness of the individuals that were carrying this trait. So we thought, why not to try to test whether CYP26 showed a similar pattern. And to test this hypothesis, one has to compare two different set of samples with two different lifestyles. And you can see here that we sampled population from two different regions of the world, because it's very important to keep the environment constant when you have to compare two different lifestyles. So here we had hunter-gatherers compared to what we will call food producer, present day, of course, hunter-gatherers, in this region of the world that we will call West Eurasia, and here we had a similar set of samples in Africa. And what we did was to sequence the whole gene, the whole CYP26 gene, including introns, so also the non-coding variants. And here you see the first result of this survey. When we translated the haplotype activity into genotype score, we observed the first difference between the two different lifestyles, whereby here you see that in food producer there is a much slower metabolism in general and the difference was significant. So hunter-gatherers seem to be faster metabolizers than food producers. Another line of evidence comes from a very traditional test of neutrality, which is Tajima's D. You will be hearing me talking about this test several times. Tajima's D is a neutrality test based on the site frequency spectrum, and is so given this definition, is affected both by selection and by demography. So if we want to test whether the signal that you have at your gene is robust in terms of selection, then you have to control for the demographic effect. And what we did here was to simply calculate the difference between the Tajima's D that we obtained for CYP26 and that we calculated for mitochondrial DNA, considering mitochondrial DNA as a neutral marker here. And we again observed that consistently in food producer, the signature was stronger and positive. But then if selection is acting, then selection is acting on phenotypes, of course. And so we wanted to see whether dividing the mutations that we found in this gene, we could see a different effect of non-synonymous mutations that are actually those mutations that really affect the phenotype. Of course, this is a rough definition, because we have heard, especially during these 2 days, that also non-coding mutation can be very important in terms of phenotype. But here we use this definition, which is kind of conservative. So when you divide mutation in non-synonymous and those mutations that should not affect the phenotype, then you see again that the Tajima's D value has consistent pattern only in the case of, or at least there is a difference between Tajima's D value for non-synonymous mutations alone, but not for the other mutations. And you see that the positive values have been observed for food producers again. So in conclusion, we had many more analysis, but I just wanted to show you those that could summarize the concept, our conclusions. So the variation of CYP2D6 might have been selected as a response to the new dietary habits during the food transition, the transition to agriculture, and the other line of evidence comes from the fact that food producers were much more similar between different populations than when hunter-gatherers were compared. And this is what you expect when there is something going on in terms of selection. And since several alleles were maintained at high frequency in each geographic region, then a plausible explanation might be that there is balance in selection in this group of individuals. Why balance in selection? A possible explanation might be that pronounced, because we know that some of this that we call slow function allele in terms of reaction to drugs have a pronounced substrate-dependent activity. So it might be that having more of them during this transition to new dietary habits could have been adaptive in some way. And similar results have been observed at other genes. You see here the example of NAT2. So this was an example of how to approach the problem, how to study the evolution going to a single gene questions. But then if now we have available many other data that come from public projects, and here I'm going to show you two different approaches again, but both on multiple genes. In the first case, I'm going to talk about Snips, and in the second case, we will see something on the sequencing of entire genes. We have seen that the signal might be very different. Why? Because here we have a certain bias, of course. So we know the Snips that before start typing, and this means that in this kind of panels we lose all the rare variants. But this kind of data set is very useful, although to try to define if there is geographic structure within our sample set. Although it's not so useful, maybe to investigate signature of selection, and for that we will move to the 1000 genome project. So here you see you see that the first approach, so I will call it multiple genes 1, that we did downloading data from the Human Genome Diversity Project. Here we have 51 populations, more than 1,000 individuals, and we concentrated our attention on 143 genes that were and we extracted from this panel about 1,000 single nucleotides. These genes were including different kind of genes important in pharmacogenetics. The first question we asked to this variation was if we could identify a specific geographic structure for these genes that was different from the rest of the genome. So in other terms, if there is a specific geographic structure for genes involved in pharmacogenetics. But how is the rest of the genome behaving? Here you see a plot. This is a very traditional way to represent genetic variation, and here is the correlation between geographic distance and genetic distance in terms of FSD, which is a measure of genetic diversity. This figure is taken from a very important paper by Ramachandran et al. Of 2005, and you see the scatter plot of FST and geographic distance at neutral loci. So you see that there is a strong correlation, which means that populations that are distant in terms of geography, they are also distant in terms of genetics. And when you do the same kind of analysis for pharmacogenetics genes, you see the same pattern. The regression is, the correlation is significant, and when you do a mantle test of matrix correlation, again, you find a significant results, which means that the pattern is very similar to what we observe for the rest of the genome, at least when we pull all the variation together. But then we wanted to see if we divide these genes in different categories, then is there any high FST or enrichment in FST in some of these categories? In other terms, some of them are more variable than others. And yes, the answer is yes, and it's very logical, as you can see here, because we found that the CYP450 genes that were enriched in molecules that that have a metabolism for foreign body compounds, they are much more variable than significantly more variable than the other others. And this is according to what has been seen in an interspecific perspective. And you see here, this is a very nice paper where these kind of genes were compared between species, and it also here it was found that genes that are important for environmental interaction, they are unstable phylogenetically speaking, so they are more variable between species than the others. But going back to the structure of this genetic variation, we applied a method that is called the APC, so the discriminant analysis of principal component, to our set of Snips. In other terms, we wanted to see whether we could identify a structure supported by this genetic variation. But when you apply these methods, then you have to know the number of clusters you want to test for beforehand. We didn't want to have an hypothesis on that. We didn't want to test for continents or anything like that. So we used a method that is called a but it's based on the k-means algorithm to ask the data to suggest us how many clusters could be supported by this genetic variation. And when we did that, we found that 6 was the most likely number of clusters. Then we used this number to run the DAPC, so interrogating the genetic variation for 6 clusters. And what we found is depicted here in these two graphs. The first one is representing the discriminant function 1 versus discriminant function 2, so the most of the genetic variation that we have in these samples of Snips. And as you can see here, the discriminant function 1 is exactly the out of Africa. So here you have populations from Africa, and on the other hand, you have populations from Europe. In other terms, this is clearly mirroring the demographic expansion out of Africa of our species. While here you see in this other panel, where you can see better represented the discriminant function 2, something that is less obvious, and it has been called by Cooper and colleague the West Eurasian sweep. And you have here populations from Africa on one hand, and populations from Europe at the other extreme. And we will see that some of these genes have been shaped like that because of selection. So now that we found that this what was the most likely representation in clusters of genetic variation at this gene, we wanted to infer how many loci and which one were actually contributing significantly to this structure. It's possible to do that by calculating the contribution of each single SNP to the structure, and then out of 1,000 SNPs, we found that only 27 contributed significantly to this representation, or at least above a certain threshold. So not many, as you can see here. So in other terms, the genetic structure that we saw is not supported by all these SNPs, but only by few of them. And here you see, I just picked up one of them, one of these SNP that is significantly contributing to the discriminant function 2, so this pattern that I called the West Eurasian sweep. And I picked up this one because there is an interesting evolutionary hypothesis behind this pattern, whereby you see here that the ancestral allele is very common and almost fixed in African population, but it becomes even the minor allele in some European population. And this has been, and the reason for this was investigated in several papers, and is the hypothesis is that there has been a selective pressure for the salt retention hypothesis, which means that this variant is associated with increasing blood pressure, because it confers a higher amount of sodium and water reuptake. So it could be adaptive in certain climate, like hot and humid climate, but it can become maladaptive once our species colonized more temperate climate. So, and that's why you see this gradient that is correlated actually with the temperature and with the distance from equator. That's the kind of question you can ask when you want to infer the structure of genetic variation, having a set of SNPs in your hand. But then if you want to infer something on selection, it's much better to have also rare variants, to have the possibility to analyze rare variation. And that's why now we have data on the 1000 Genome Project. We can use this data exactly on that purpose. So again, we downloaded the genetic variation on 117 genes. This time, we concentrated our attention on what we think might be interesting in pharmacogenetic terms. So we used this gene drugs set of samples coming from this CPIC project. The distribution of the population now is this one that you see, is 26 populations. And the number of SNPs that we analyze is here. So now that we had all these variation, including rare variants, we still wanted to test whether the pharmacogenetics gene were more variable than neutral loci. And as you can see here, you have in blue, you have the neutral loci. In orange, you have the set of samples that we are analyzing, including HLA, which is probably the reason why here the heterozygosity is so high. But when you remove the other genes and you concentrate your attention only on the drug metabolizing enzyme, again, the variation is much higher than what you observe for neutral markers. So this didn't change, including rare variants. Next step was to try to infer, that is very popular now, to try to infer if there is a positive selection by local adaptation. And one way to do that is to use these methods that are called FST outliers. So you want to know whether you interrogate each dot here is a SNP, and the methods are based on the relationship between genetic distance conditional on heterozygosity. What are you doing? Basically, you are calculating confidence interval based on coalescent simulation or trained by neutral data. We used neutral data here. And then you find SNPs that are outlier in this pattern. So those that have a higher than expected amount of genetic diversity conditional on the level of heterozygosity. Those loci, those SNPs are those that are showing signature of local adaptation. We found an incredible amount of genes here, 60 out of 117. So this is not really a nice number. I would say we will have to look into these genes because the number is too high. But not that this would be false positive, but just that this test might be sensitive also to other factors here. But then when we looked into this gene, most of these variations were intronic, and but only 5 SNPs were linked to drug response. So at this, we are still analyzing the data, but we concentrated our first attention on this CYP, 5 SNPs involved in drug response. The pattern that you observe when there is this local adaptation is something like this. So you have one SNP very frequent in some population and exactly the other variant that increases very much in frequency in the other region of the world. In these genes that showed signature of local adaptation, we found those that I'm calling here the known cases of CYP3A clusters, and we will see another case that was already known for positive selection. The CYP3A cluster, here you have this variant, that is the one that I showed you before, and again, the reason why probably this variant has been selected is again in the framework of this SOLST sensitivity hypothesis, so somehow correlated with the temperature. But then again, another gene that we found showing signature of local adaptation was this VUK-ORC1, and here the signature is less clear because actually the geographic pattern is very clear because this is exactly what you expect when there is local adaptation. So only certain populations show a very high frequency of the derived variant. But the reason why there has been this selective pressure is, in my opinion, is not so clear, although in this paper they claim that it is. But clearly this gene can be selected and is important for coagulation. And you see the example of artificial selection, actually, because here you see the example of the mice where by introgression a species of mice became resistant to warfarin that was given as a poison, and just when this gene got introgressed from another species, then the frequency of this haplotype increased very much in frequency due to selection because of these poisonous substances. So it's plausible that there is something in the environment that pushed the frequency of the derived variant up, but still it's not, the reason is not so clear. So apart from these two known cases, we wanted to see whether, we wanted to somehow infer another kind of signature of selection, because positive selection is very, like I said, is very popular, is exciting to find signature of something that changed in the genome and try to find the reason why. But most of our genome is showing signature of conservation. And and that's the reason why we saw in the previous talk that the frequency of the non-synonymous variant is so much lower than the rest of the variants, because there is conservation, because there is selective constraint, especially on the genes that are coding for important protein. And this is what we've observed here. So again, we applied the Tajima's D to our data set. We calculated this test of neutrality for each of these loci for each of this population. And you see here the distribution of the value of the Tajima's D test. And you see that these are the extreme values, so are those that are giving us the signature of that something is going on in terms of selection. The neutral, the expectation is 0 when there is neutral evolution. Here we have the negative values that are indicating usually signature of directional selection or population expansion, because like I mentioned, this test is affected also by demography. And those genes that were showing signature here were those that I mentioned already because of positive selection, but then we also had some other signature. You see here that VUK-ORC1 was showing different values of Tajima's D. Some of them were extreme, and because of that local adaptation, but in some other population there was no signature. And similarly, we saw something, the trend for CYP3A5. But then you see here that PROS1 stood up, was evident because in this protein, the signature given by Tajima's D was consistently negative for all populations, which is what you expect when there is purifying selection acting on a gene. And this is not surprising because there is an OMIM entry for this gene because it's correlated with there is a dominant variant that is correlated to thrombophilia. So just to conclude, we found with this analysis, we found that signature of selective pressure for sure on the coagulation pathway, and this was very interesting for us because we saw signature on this gene and on the other gene, different signature, and they were given by, this is the pattern that you observe when there is positive selection due to local adaptation, and this is the pattern that you observe when there is selective constraint. In other terms, you see that only very rare variants are maintained in the population. Purifying selection is sort of cleaning for them. And so this, in the end, we concluded that this is an interesting pathway that we might keep on studying in the next step. So what we are going to do with this data, again, we are going to look for correlation with environmental variables. Now it is very easy using GPS data to correlate the genetic variation that you see with the distribution of different environmental variables, and which are telling you something of course, on the environment and are possibly shaping the evolution of the gene. We are undertaking interspecific analysis using ancient genome that now are available, so Neanderthal, Denisova, and other non-human primates. And then, given the signature that we observe on coagulation, we are going for sure to do some network analysis on specific metabolic pathway, like the one that you see here. Okay, so, and with this, I think I'm finished, and I really want to thank the people from Ferrara that are helping me in these studies, and Johanna Sistonen, who is here in the audience for the work on CYP36, and Pierpaolo Maisano-Delser for the work on the genetic structure. And thank you all for your attention.
Thank you, Son. Yeah, thank you again, Silvia, and thank you again for traveling such a long distance to be with us today. We have time for maybe 1 or 2 questions. you have a question?
Yeah, very interesting. So I always thought about, if I look at the CYP genes, that there's evidence for positive selection for loss of function mutations and for gain of function mutations. And so I was thinking that maybe the reason is that combined, all CYP genes combined, CYP genes combined in the liver are kind of saturated. So there's already so much protein in there that if a new chemical toxin were to come, you cannot, upregulate a gene unless you downregulate another gene. So have you thought about that possibility, and how would that play out in the data when you look at multiple genes in that in the CYP family?
I didn't look at the CYP. Are you mentioning the CYP3A genes or CYP in general?
All CYPs together, because the liver is, it has the color because this is one of the major proteins in there, and it's probably saturated. You can't put much more in.
Yes. This is the kind of feedback that we need for people working in your field, because we look at signature, but then we need interpretation. So it's very nice to have this kind of information. But the point is that at least for what I see, each different single CYP gene is behaving very differently. So I don't see a combined signature, and the signature for positive selection is evident only for CYP3A4 and CYP3A5. The other CYP don't show signature. No, there's, at least as far as I know, there's no reason for.
Okay, one more.
Sylvia over here. Yes, really nice talk. So could you just comment a little bit more on what you plan to do with the ancient humans that have been found, and how you plan to relate their genotypes with perhaps learning something about our ancestors?
Sure. Genotype is a big word because, as you know, the coverage is not so high in some cases. So, or at least with ancient DNA, it's difficult to have genotypes. We usually have one sort of consensus sequence. The idea is to check whether the variants that are present in humans, or the most common variant, are also present in ancient genome. And for example, in my group, there has been, they just published a paper on the Euromoduling, where they see that the variant that is common in humans is not the one that is common in these ancient species. And so you can draw conclusion on the evolution of the genes depending really on the pathway they are involved in, considering the history of these different species. And but in an interspecific perspective, you can also kind of use this test like that those that are non-synonymous versus synonymous mutation to infer whether there's been a signature towards certain specific kind of selection.